Hello, I'm called Atwine Ambrose. I'm going to demonstrate a technique called membrane filtration technique. It's used in water analysis, and particularly we are going to look at E. coli and other coliforms, and the media we use is called E. coli coliforms chromogenic agar. This is a differential medium. It can be able to grow both E. coli and other coliforms, but then you are able to differentiate between E. coli and other coliforms after incubation based on the color of the colonies. And now, before we do our analysis, we have to make sure that we have everything we need within our proximity. And here with me, I have plates of ECA, which is E. coli coliforms chromogenic agar. And these plates, since I want to do in duplicates, I have 2 plates. And then I have my water sample. This is the water sample I want to analyze. I have sterile water that I will use later. Then I have the diluent that I want to use as I analyze my 1 ml. And then I have my filtration bridge. So before we start the analysis, we have to make sure that everything we use is sterile. And one of them is the membrane filtration unit, which we have to sterilize. And then it has 2 ways of sterilization. One, you can sterilize in an autoclave at 121 degrees Celsius for 15 minutes, or you can as well use absolute alcohol, just pour in some little, and then you set it ablaze. I will demonstrate how to sterilize my membrane filtration bridge. I will use absolute ethanol. I pour in a little. After pouring in my ethanol, then I'll use the pair of forceps to put in the flame. So now my membrane filtration bridge has been set ablaze for sterilization. So I'll wait for the flame is out. I'll use cold sterile water to quickly cool down the filtration bridge. So when the sterile water is done running through the bridge, now I'm ready to analyze my sample. Here I have membrane filters, which I will use in this analysis. Using a pair of forceps, I will pick a membrane filter to place on the bridge for analysis. But before that, I'll first make sure my forceps are sterile using absolute ethanol. So I'll wait for my forceps to cool down. Now I can be able to pick my filter. When opened, has 2 pieces of paper, but one of them is the filter that I want to use. It has grids. So I'll pick the one that has grids. I press on the bridge. Now that I want to use 2 funnels, I will press 2 filters. So now I open and pick the second filter. So as you pick, make sure you avoid touching the filter. Now, after placing the membrane filters on the bridge, now I'm ready to analyze my sample. But before analysis, I have to shake well to make sure that the sample is well homogenized. Then, since I want to analyze 100 and then 1 ml, because I am not sure of the status of the sample, the contamination status, I have to make sure I do different volumes so that after incubation, I'm able to get countable colonies. Otherwise, if you only do 1 volume, say 100, you might get a lot of counts which you will not be able to differentiate. So, I will start by pouring in 100 mils. This is where I'm going to put my 100 mils. And since the funnel is graduated, I can be able to power up to the mark. Now I'm going to put in my 1 ml, but since 1 ml is so small, I will not be able to filter it without putting it in a diluent. Here I have a bottle of sterile diluent. So I will pick 1 ml from my sample. Then I will homogenize the 1 ml within the diluent. Then I can now power in the next funnel. So from here I open the taps and I switch on the vacuum pump. So I can close the funnel for the 1 ml since it has already gotten done. I close the funnel for the 100 ml and I switch off the pump. So from here, now I'm ready to pick the filter papers from the funnel and pressing them on my agar plates. As I mentioned, I have an agar plate where I'm going to place the filter paper through which I've filtered 100 ml, and then this is where I'm going to place the filter paper for the 1 ml. I have to make sure the pair of forceps is sterile before I pick. This is where I'm going to put as I place the filter paper on the agar plate. I make sure I place it very well to make sure there's no air bubble left in between the agar and the paper. Now that I'm going to pick the next then I have to also sterilize the forceps again, and then use So after placing the filter paper on the agar, now the plates are ready for intubation. Please note that you should always replicate the analysis, and when placing the plates into the incubator, you should place them when they are upright. Otherwise, when you press them upside down, the filter paper might fall on the cover. And then when you are going to work on the next sample, remember to re-sterilize the membrane filtration bridge, because otherwise it will contaminate the next sample. After 24 hours of incubation at 37 degrees Celsius, then we have to analyze our results. And here, these are our plates. They have pink colonies and blue colonies. And now that we used E. coli coliforms chromogenic agar, which is a differential medium, it is able to grow both E. coli and other coliforms, but then differentiate them in terms of color. E. coli appear as blue colonies, and then other coliforms appear as pink. And now that we filtered 100 mils and 1 mil, you realize that where we filtered 100 mils, the colonies are too many. You can't be able to count. But where we filtered 1 mil, you are able to count. And now, looking at the plate where we have 1 mil, I can see the counts are around 100. So if you want to report per 100 mils, because most standards want us to report results per 100 mils when we used this technique of membrane filtration. So if in 1 mil I got 100 CFUs, so what about 100 mils? So I multiply. So in 100 mils, I will be having 10,000 CFUs per 100 mils. So you realize you can't be able to count, even if you are given a plate which has 10,000 colonies, you can't be able to count. But you see, plating 1 mil has enabled us to be able to estimate the original E. coli and coliforms in the sample. And looking at the standard of water, they don't recommend water which has E. coli to be consumed by people, given the health effects and other effects that might come. And now looking at a sample which has 10,000 CFUs per 100 mils, that one is already too contaminated for human consumption. Note, the presence of blue colonies gives us some confidence that the sample had E. coli, but there are other further tests you can do to confirm and be sure that there was E. coli. For example, you can do biochemical tests, you can do genetic tests. There you will be able to confirm E. coli and or even other pathogenic strains of E. coli. And more so, there are other media that you can use in analysis of E. coli, apart from E. coli coliforms chromogenic agar.